hopefully they can get the presentation started for my
Yeah,
part.
1 second, Charlie.
Okay,
We
I'll
just
wait.
need his up on screen presentation. No, that's it. The second one. Yep, Alpers. Yep, number 2. Yep. Yep.
Okay, great, thank you. So I want to start by thanking the organizers of the symposium. It's a fantastic opportunity for those of us working in the Clear Lake area to share our knowledge and our preliminary results, and to network with everybody and reach a wider audience for our science. I'm Charlie Alpers with the U.S. Geological Survey, working out of the Sacramento office, the California Water Science Center, and this presentation has a large number of co-authors. I'm not going to mention everybody, but the ones in green are from USGS. The ones in various shades of blue are from the University of California. We're working with folks from 3 different campuses, Santa Cruz, Merced, and Davis. And we're also working with scientists from Stanford at the Linear Accelerator, and Reed Harris, a consultant from Canada, who's working with the modeling that you're going to hear about in the next presentation by Sergio Valbuena from UC Davis. Funding for this project was primarily from US EPA through Carter's management of the Superfund site. There's some work on mercury is also funded through the Blue Ribbon Committee and the California Natural Resources Agency, mostly on the tributaries to Clear Lake. And I'm not really going to talk about that much in this presentation. They're also funding the mercury modeling that Sergio is going to talk about. And then the state funding has partial match from the USGS Cooperative Water Program. Here's an outline of my presentation. I'll spend a little bit of time talking about project goals and the conceptual model of mercury cycling. Then I'm going to get into our study approach, which is divided into work at the mine site in the areas you see there, and then work in Clear Lake. I'll point out that there's a poster tonight also about fish mercury concentrations that some of you may have a keen interest in, and so there'll be more information. I may have to go through that part pretty quickly since we're running late. The last part of the scientific part of the presentation is on mercury isotopes. One of the goals of our study, as you'll see, was to characterize, use that tool of mercury isotopes, relatively new tool in the scientific toolbox, to evaluate what's the source of mercury in the food web of Clear Lake. Is it really all coming from the mine? Are there any other sources we can identify through the use of mercury isotopes? So all of the other parts of the study kind of feed into that. Samples that we've taken along the way at the mine site and in the lake are taken with the idea of analyzing mercury isotopes to try to address that problem, and then I'll wrap up with some summary statements at the end. So in terms of the goals, we're really trying to support EPA in terms of determining possible benefits of their remedial actions at the site. Trying to understand the interactions between contaminants from the site and the sediment water fish in Clear Lake. And again, Carter mentioned methyl mercury. It's the organic form of mercury that does bioaccumulate. It's more toxic than the inorganic mercury, and it's the part that bioaccumulates, so it's very important to identify how that methyl mercury forms. Can that part of the cycle be disrupted? Because the mercury is already in Clear Lake, but can we keep it out of the fish by maybe interrupting that methylation cycle? And so understanding more of the details of the pathways are important. And again, using the mercury isotopes to try to quantify and identify mercury sources. And then finally, the calibration of the model that I mentioned, the 3D hydrodynamic model that UC Davis has developed and that Sergio is now extending to include mercury. The data we're collecting will be useful in calibrating that model. And there's going to be a separate mercury bioaccumulation model that's not part of the 3D part, but it'll be it's something else that we're working on, and that all the biological data will help feed that model. So I got a sequence of sort of cartoons here about the mercury cycle. I'm going to spend a little time on this just to get people familiar. I think many people in the room probably aren't familiar with all the ins and outs of mercury, and I won't have time to go into great detail, but I want to make a few points about it. I don't know if this has a pointer. I'll just use my finger. Those are intended to indicate like head frames on an underground mine. Of course, there can be surface mines. So mercury mines are a source of mercury contamination. The red color is where there's high mercury contamination, and the other colors are less and less mercury contamination. There's naturally rich, enriched soils. There's also hot springs, which we do have at Sulphur Bank Mine. You can think of Sulphur Bank as kind of a mini Yellowstone. It's got fumaroles. It's got hydrogen sulfide gas coming out. There's mercury gas coming out, which is also true at Yellowstone. And there's, as Carter mentioned, there's a mercury deposit actively being formed. The whole thing is only about 40,000 years old. It's a young geologic feature. And all the volcanics around here, as you'll probably hear tomorrow from Seth Blatter's talk for the USGS on the geology of the Clear Lake volcanics, the whole formation is only 2 million years old, which is very young geologically. And this mine is only 40,000 years old or less, very, very young. And so the mercury sulfide, HGS, is sort of the mineral cinnabar, is the ore mineral. It'd be pretty red color you've probably seen. And artists even used it as a pigment, unfortunately, for the ones who lick their brushes. The mercury 2 is an oxidized form of mercury that is kind of a precursor to the methyl mercury. That CH3Hg plus is the formula for methyl mercury. So there's back and forth arrows between all those because microbial reactions can affect which form the mercury is in. So it's very complicated with microbes, and then there's also abiotic reactions that affect all this. At the very top of the screen, it got a little messed up, but Hg0 can also, the gas can go into the atmosphere. Hg0 is a liquid at room temperature, but even solid phases like cinnabar lose gaseous mercury to the environment, especially on a hot day. They're exposed on the waste rock piles. So these calcines, or the leftovers from the retort process where they heated up the ore to extract the mercury, those are relatively low in mercury, but they do have different forms such as sulfates and chlorides that are more soluble in addition to elemental mercury and the original cinnabar. When you get off-site and get in and think about that as being Clear Lake, you get transport, as Carter mentioned, they just dumped mine waste into Clear Lake. So you've got all these contaminated sediments at the bottom. You can have methylation in the subsurface and demethylation. And then once the methyl mercury gets into the food web, it gets into phytoplankton or algae, and then it gets, and then the whole food chain starts. You got zooplankton eating the phytoplankton, you got small fish and invertebrates eating the zooplankton, and then large fish eating the small fish. And one unique feature about mercury, very few metals do this, it biomagnifies up the food chain. So every step you go up the food chain, the concentration of methyl mercury is 3 to 5 times higher. So we're actually looking at parts per trillion in the water, parts per billion in the algae, and parts per million in the fish. It goes up by 1 million times between the water and the fish. That's called a bioaccumulation factor. So it's a very complicated system, and the details of the food web matter. There's atmospheric exchange that I won't be able to get into, but this is a very simplified version of the mercury cycle. There's a lot going on. And then on top of it all, I said there's methylation in the subsurface. The microbes do that. Generally, the microbes that change the forms of sulfur and iron, they're called sulfur and iron reducing bacteria. They can methylate mercury. They can also do their thing in the lower part of the water column. When the water goes anoxic, as was mentioned, anoxic water can lead to phosphorus release from the lake bed. It can also lead to methyl mercury formation in the water column itself, which is an important factor. Okay, so we're going to now talk about the different studies we've been doing at the mine site: mine waste, groundwater, air, and lichen. You saw similar maps to this from Carter. The waste piles are shown in purple. The tailings, or so-called calcines, are where we have the orange triangles. We sampled those in the red area. And there were actually some areas mapped as ore piles shown in the green with the red triangles. And those had some of the highest concentrations of mercury that we collected, up to maybe a weight % mercury in this leftover ore. The orange squares at the bottom where they actually did the processing, the retort facilities, or those buildings. And there's mercury in the soil there from that. So we took 15 samples of composites in these waste rock piles, plus 6 drill cores from the waste rock dam area here. And those 21 samples have been characterized by a number of methods shown here. I won't go into them all. The ones in purple I'm not going to hardly mention at all. The ones in green I'll say very briefly about, looking at metals, mercury and sulfur isotopes, and then the speciation of mercury using some pretty sophisticated technologies. I mean, Carter worried you about alphabet soup. That's one of the worst ones there. But I'll explain what some of these acronyms are on a subsequent slide. This is we used an instrument called the portable X-ray fluorescence when we're out in the field. Just give us a quick idea of what the mercury, arsenic, and antimony concentrations are. And it turns out when we analyze them in the laboratory with inductively coupled plasma, sorry, another acronym for ICP, the concentrations are very similar to this, but those big red circles, there's lots of mercury in the ore piles. The smaller multicolored circles in between are the calcine tailings, where the concentrations are quite a bit lower. They've removed the mercury from that stuff, but they didn't get it all out. It's still on the order of 100 ppm, which is still way above background, but it's not thousands of ppm. But just for reference, 10,000 ppm is 1%. So 5,000 ppm is 0.5%. And milligrams per kilogram is the same thing as ppm. So these pie charts show you the arsenic in gray and the antimony in brown, and the mercury in red. So it's pretty much all three metals are everywhere, but there's particularly hotspots for mercury in the ore piles and some of the waste piles. Here's just some photographs of what the mercury ore looks like, some cinnabar colored material, but iron oxides can also be that color, so it can be a little tricky. That's not all solid mercury. The tailings are bright red because the iron transforms to hematite. And then here's what some of the waste rock looks like. This is an exposure in Muir Clear Lake where it's been covered with some other soils because some of this was deposited, decades ago. Here's a quick tour of the synchrotron facility at Stanford where we're doing some very sophisticated work with our partners at UC Merced and the SLAC Linear Accelerator. I won't go into the details, but there's what the acronyms stand for if you care to read those. It's a sophisticated technique that has become even more sophisticated in recent years. There's a new detector that's high resolution for this Zane spectroscopy. So the blue and green were the old spectra, and with the new high resolution, we've got the red and the purple spectrum, much more featured and much better for figuring out what's in these mixed materials. And there's two forms of mercury sulfide, cinnabar and metacinnabar. We can distinguish those with this technique. And then here's an example of actual mine waste on the top and the fit to these. The blue is the real spectra, the red's the fit, and we've got some of both kinds of cinnabar, and then we've got a little bit of mercury sulfate in this particular sample, which had 1,000 ppm. We have reference compounds of all those minerals you see on the right and a few inorganic species. So we have a very nice library associated with it. And this is a collaboration with Peggy O'Day, the professor at UC Merced, and one of her grad students. I'm not going to talk about the stuff in purple, except to say we've done some extensive work. Carter mentioned the groundwater study with those flow lines going from permanent impoundment to Clear Lake. USGS has been helping EPA with interpretation of sulfur and oxygen isotopes in the sulfate, which forms when the acid water forms. Acid rock drainage is basically pyrite or marcasite, iron sulfide turning into sulfate and lowering the pH and increasing the acidity. Those isotopes plus the isotopes in water can be very diagnostic for areas. Where's the water? Where's the sulfate coming from? Trying to tease apart what's really going on in those reactions in the waste rock dam. CO2 flux has been mapped by one of my colleagues with the California Volcano Observatory, Jen Lewicki. She did extensive mapping of CO2 on the site. There's a lot of CO2 fluxing out from the hydrothermal geothermal system, and it can help figure out where fractures are. So that's the stuff I'm not talking about. The blue are the H2S from vents. So I'll just have one slide of sulfur isotopes. Mercury and air, we've done some monitoring of that. I'll show you briefly. And then the lichen work by UC Santa Cruz. Peter Weiss-Penzias is here in the audience from UC Santa Cruz. His group's done some really cool work showing how lichen, which grow naturally on the trees out there, can be used as integrators of the gaseous mercury. And that can help understand spatial variations rather inexpensively. Here's Sarah Peak from the USGS Volcano Group sampling H2S at one of the wells. There's our H2S meter saying 78 ppm, which is hazardous. And that's a level you definitely don't want to breathe. And here's a picture of some sulfur crystals that occur naturally and some sulfate minerals you see on the surface. The sulfur isotopes, most of them are near that 0 in the middle. The units are per mil, which means parts per thousand relative to a meteorite. That's sort of the average abundance in the Earth is pretty much near 0. But that green box for marcasite's another form of pyrite basically. That's what leads to a lot of the acid rock drainage, and we're seeing some of the groundwater show that negative numbers in sulfur isotopes telling us that's where that's coming from. So it's very, very useful tracer for acid rock drainage. Here's some pictures of Peter Weiss-Penzias's lichen samples. I don't have time to go into much detail about this, but they've looked at several species. They're pretty consistent between species. He's looked at some hot spots at the mine. He's also looked at some control sites around the lake, including one right here in Nice, and another one in Ukiah. So you have the upper part of the map. You can see where we are on the map. There in Nice. There's a sample in Ukiah, and some others around the lake that are in the yellow and orange colors. Those are relatively low in mercury, and then at the mine site, it's blown up here on the left. All the red dots and orange dots show much higher concentrations in the lichens, and we're getting into detail about not only the concentrations, but also what form is the mercury in the lichen can be helpful. We did some air sampling at the mine site every 6 weeks. We did a collection of a device that's been out there for a 2-week period. So this is kind of a time series, and there's 2 sites at Nice and Ukiah down at the bottom with 1 nanogram per cubic meter. That's how much air passes through our collection device. And the black dots are from the mine site where we have substantially higher mercury concentrations. Not surprising, but we were able to quantify that and see what the annual cycle looks like. These measurements have been continuing for a full year. Previous studies about 20 years ago by a group out of University of Nevada, Reno showed quite high fluxes of mercury out of the mine waste. There's a wide range, but some of these numbers are the highest recorded almost anywhere in the world. And it leads us to think that, they calculated that 8 kilograms a year of mercury are going into the vapor from the mine, and that's a lot more than is going through the waste rock dam, but we don't know where that vapor goes. Some of it could be deposited by either wet or dry deposition in the nearby watershed, such as Schindler Creek. That's going to be a future investigation. Okay, we're done with the mine site. I know we're running late on time, so I'm going to go on to the Clear Lake characterization. We're going to talk about sediment and pore water on the bottom of Clear Lake, some water column work, and the work on the biota, which again I'll go through quickly because you'll be able to see that also at the poster session. First with sediment, we partnered with UC Davis. They took some cores back in 2019 as part of a study. In fact, Mika Swan is here, sitting next to Jeff Schladow there. Mika's work took these cores and subjected them to both oxidized and reducing conditions as an experiment to see if they would release phosphorus under reducing conditions, which they did. Over on the right is just mercury down the cores, and there's units of centimeters as you go down to 20 centimeters. It shows how it's pretty consistent in terms of concentration. The Oaks Arm in green had the highest concentrations, over 40,000. That's ppb, so it's 40 ppm in the Oaks Arm, and then still, upwards of 10 ppm in the Narrows, that red circle between the Oaks Arm and the upper arm. And in fact, the whole lake is contaminated, as Carter said, because of the currents. And it's thought that most of that mercury, if not all of it, comes from Sulfur Bank Mercury Mine. And so again, the isotopes are intended to address that. So this is what we had to start with. We looked at the top 2 centimeters of these cores that Mika subjected to these oxidation and reduction experiments. And total mercury was basically unchanged, and that's not a significant change. But reactive mercury, the oxidized form, increased in the oxy treatments and was lower in the anoxic treatments, as we'd expect. And the methyl mercury, wait for it, here we go, increased in the anoxic experiments and decreased in the oxic, and that was also a significant difference. So this is very instructive. It confirms what we recognize to be the case, that methyl mercury forms in reducing conditions. These are just a couple maps I'm going to show of concentrations of mercury in some of our sediment samples, obviously very high near Sulfur Bank Mine, which is over at the east end of the Oaks Arm, and lower in the rest of the lake. And then here's methyl mercury, also the similar pattern, very much higher in the Oaks Arm. I'll go back and just show that we also measured a few different things in the sediments. We looked at organic content, sulfur, and iron chemistry as well, but I'm not going to have time to talk about those things. But those are helpful because we're trying to track the activity of these microbes that that methylate mercury. If you look at iron and sulfur chemistry, it helps us understand how active those microbes are. I'm trying to advance here. But going, okay, one more. Okay, Carter actually already showed this map. It shows our water sampling locations in all 3 arms of the lake. Again, the long list of constituents that we're measuring. On the top are the ones we measure in the field with a device called an XO2. It's a sonde. And I want to just highlight, these are the sort of standard water quality parameters, but fluorescent dissolved organic matter, or FDOM, may be something people aren't that familiar with. It's actually a really handy thing to measure because it correlates with methyl mercury. So we might have a chance, just like turbidity correlates with suspended sediment, we're actively working on developing what we call proxies to use that FDOM plus some other fluorescent indicators to get a real-time continuous measurements of methyl mercury and total mercury, as it turns out. So that's a pretty exciting direction for this research. We're measuring all these different forms of mercury that are shown, suspended sediments, chlorophyll, various forms of nitrogen and phosphorus, which are partly to benefit the UC Davis work in the lake, dissolved organic carbon. And again, this optical properties, or EEMS, I'll show some detail about that. That includes the FDOM plus a bunch of other things that can help us with that research on proxies. Oh, I might have lost the whole thing here. We'll see. So when it comes to using that information for proxies, one of the ideas is to, okay, I'm back. I'll get to the proxy in a second. I just want to show I showed that there's temporal variation of methyl mercury. It's higher in the Oaks Arm. There's also, spatial variation of methyl. This is temporal variation of methyl. It's much higher in summer of 2021 when we had a major anoxic event in the lake. We got methyl mercury concentrations about 10 times higher, which is, again, what we expect, but we need this kind of data to calibrate that model of how fast does it happen and exactly where does it happen. Okay. And this is just the first couple of years of our data. We have other data from 22 and 23, and we'll be doing more of this kind of sampling in the future. Oh boy. So the next slide shows the EAM spectra. It's a very colorful slide, as you can see. And well, it skipped it. I want to show my other colorful slide. Come on. It's not letting me. There it is. Okay. I promised a colorful slide. So the x-axis is excitation, and the y-axis is emission. They're called, and that's why it's called an EAM spectra, excitation emission matrix. And this isn't actually the raw data. It's the correlation of two different forms of mercury with the EAM spectra. It's a very sophisticated measurement of organic matter quality. And FDOM is labeled here. This rectangle is the commercially available FDOM. And there's actually, you can't really see it, but there's a white one in the background. Different companies make a different FDOM. It's not just all the same thing. You have to be very specific about which wavelengths you're looking at. But anyway, we're using FDOM. It correlates with methyl mercury because that's a bright part of the diagram. This is on the left is whole water methyl. On the right, filtered methyl correlates with a different part of the diagram. And we're actually going to design a custom probe to measure that red square so that we can, you know, come up with something that works even better for filtered methyl. So this is ongoing work. USGS does this kind of work around the country. We're using it for PFAS, which you may have heard of as another emerging contaminant. And it's a very exciting technology to use this probe. This is a company called Turner makes this unit with 5 different fluorescence probes. And we can customize these for each water body to try to get correlations and use this as a proxy. If this actually works, we can get a reading every 15 minutes. A methyl mercury concentration costs several $100s each. So you can imagine how much valuable data you're getting just even in a day. Several $100s for a methyl mercury analysis. Probably 150 net with overhead about 300.
To do it the old-fashioned way.
Yeah.
It's hundreds of dollars a sample. To do it this way, you get a measurement every 15 minutes.
Just for the, yeah, so we wouldn't, yeah, anyway, it saves money and it gives you a ton more data and you get the temporal variation when it goes anoxic. How long does it take to get the methyl mercury to build up? So it's a really exciting opportunity. Okay, we're going to transition to biota here in the interest of time. Our group out of Corvallis, Oregon, led by Colin Eaglesmith. Colin actually was part of the team that worked on Clear Lake back in the 1990s and early 2000s. I'll show the reference in a minute. He worked with a guy named Tom Sakanak, who worked at UC Davis and then U.S. Fish and Wildlife Service and then USGS, and is now retired but still affiliated with UC Davis. Tom did a tremendous amount of work with his group, and Colin was part of that group, and he's very excited to be back working at Clear Lake again later in his career. And Colin himself is a very well-established mercury scientist now, working all over the country. And his group has done a fantastic job of collecting not only fish, but also zooplankton and benthic invertebrates, shallow and deep. This map is just from their first year of collection in this project in summer 2020, but it shows how they've done zooplankton, the blue dots, pretty much all around the lake. They've got fish on the shallow areas where they tend to get the smaller fish, and then also they did some e-fishing, and then they did the deep benthic samples as well as some shallow benthic samples. So they've done a very comprehensive job on the biota sampling, and it's gone on from basically for 4 years, 2020, 21, 22, and 23. I'm going to show data just from the first 3 years of that. And I'm not going to go into detail, but this is just the fish data, and it's broken down by species and also sizes, because as fish get bigger, they get bigger mouths and they can eat bigger prey, and then the biomagnification happens, and the higher trophic level, which means, you know, the ones at the top of the food chain have a high trophic level, they always have higher mercury than the lower trophic level or the smaller fish of that same species. And it shows how for every color you see it larger in the Oaks arm. This is over 1,800 fish that we collected in those 4 years. And so the pattern is quite clear, but it's easier to look at it one species at a time. But this kind of gives you the big picture that the Oaks arm's at least twice as big in terms of mercury as some of the other arms, and it's true for just about every species of fish. And I lost the video again, but I've got some other really colorful graphs coming up that you'll enjoy. Okay. And some pictures of fish. Everyone likes that. We skipped one. I think largemouth bass is very shy today, apparently. But anyway, there's a largemouth bass. That picture's not from Clear Lake, but they're very popular for fishing. Clear Lake's a bass destination. I understand there's tournaments out here, and it's largely catch and release, but you know, you can see the Oaks arm in orange has about twice as much mercury as the other arms. And I'll point out that the horizontal scale is linear, but the vertical scale is logarithmic, which means each tick is 10 times higher, 10, 100, 1,000. And these units are parts per billion in the fish, wet weight. And that's people generally eat wet fish, basically, although some people eat dried fish, I understand, especially tribal practice to eat dried fish. So you need to keep that in mind when you look at the data. Is it wet or dry? Because there's a difference of about a factor of 5. The dry fish is 5 times more concentrated. So we put it in terms of wet weight because that's how the fish advisories are written. The state agency in charge of fish advisories is called OEHA, O-E-H-H-A. If you want to learn about fish consumption advisories in Clear Lake, go to OEHA's website. It's oehha.ca.gov. Thank you for catching me up again. I'll skip the bass and talk about bluegill. You see the same thing where the oaks arm is higher, but we always look at as a function of size, which is also the age of the fish, and weight pretty much correlates with length, but length is typically used. And in this one, the orange slope's a little off. We don't have very many large bluegill for this particular data analysis. And then we also have data for 2 other species, if I can get to them, technically. Here's black crappie. Similarly, for the small ones, it's very clear the oaks arm has higher concentrations. And for the limited data we have in the larger ones as well. And then finally for silver sides, this is the silver sides on the next slide is what we consider a prey fish. It's a small fish. Oh. I didn't do it. Silver sides are a tiny little fish. The one that's mapped here is called Mississippi silver sides. It's actually an invasive species, but it's the one that's out there now. Anyway, it's a very small fish, and they're very sensitive year to year to mercury concentrations. The next slide after that one that showed that silver slides are higher in the oaks arm shows that interannual variation in 2021, where we had that very large anoxic event with high methylmercury in the water that I showed earlier, we had higher methylmercury in the zooplankton and the silver sides, where it was about half as much in 2020 and 2022. So we see that these small fish and the zooplankton respond quite quickly to the changing environment out there, and we think that they're going to be very good species for monitoring. Actually, we're designing the monitoring for the hydrolytic oxidation system, HOS. Those are going to be key species for looking at that. So we're now skipping to mercury isotopes, apparently, which is good, because we need to move along here. Okay, next, please. There was another summary slide on bio. Okay, let's look at this. This is also on the poster, so if you can come to the poster session, you can have a closer look at this. I don't have a lot of time to talk about it, but the slide on the right was published by Tom Sakanik and others in 2008. Again, an excellent compilation of papers in the journal Ecological Applications, including my co-author on this paper, Colin Eaglesmith. And they analyzed over 1,000 fish and compiled data all the way back to 1976. And you can see those diagonal lines going up to the right as mercury increases with larger fish. The red line is for bass. The green line is for silver sides. They call them inland silver sides. They're now known as Mississippi silver sides. And there's a few other species in there. But again, the pattern is really quite similar. We tried to plot it up with the same scale and the same colors. And you can see that, long term, over decades, the mercury patterns in fish are pretty similar. We haven't done a real in-depth quantitative analysis yet, but on the face of it, the bass line looks pretty similar. There may be a few bass over 1 ppm, which we have not seen in our study yet, so we can't really make a case that things are lower, but for the data we have, the spatial patterns look pretty similar. And again, there are changes from year to year. We've been studying over 4 years. That previous work was done over 20 years, 25, almost 30 years. So it takes a lot of work to kind of tease out if there are any long-term trends. But it's important to have data to make that evaluation. And we have over 1,200 fish in these first 3 years, and we'll have another large number coming up that we sampled in 2023. Okay, again, I'm happy to talk about this in detail at the poster session with anybody who wants to come by. Although I'm not a biologist, I'll just say my colleagues, John Pierce and Colin Eaglesmith, are the experts on this. Okay, now we're going to go to mercury isotopes. And this is the last technical part of the talk. Some of you may not even know what an isotope is. It's basically a different mass number of the same element. You've probably heard of heavy water or hydrogen 2 that has an extra neutron in its nucleus. It's still hydrogen because it has 1 proton, but if it has a neutron also, it's deuterium. It's a tube mass. Well, the same thing with mercury. There's 7 different naturally occurring isotopes, and they all have the same number of protons, but they have increasing numbers of neutrons. And so the natural abundance percentages are well known, and you can measure all of these on an instrument called the mass spectrometer that you see there at the top. And mercury's one of the elements that has a pretty complicated behavior. We do all this ratioing and comparing it to standards and stuff like that. So I'm not going to go through the equations on the bottom, but what you get out of it are these numbers we call deltas. And a small delta, like that top one that says delta 202 Hg, Hg of course is symbol for mercury, that experiences what's called mass-dependent fractionation. And it's useful for tracking the source of mercury. Those ratios don't change no matter what you do to when you cycle it in the environment, you methylate it, you oxidize it, reduce it, that pretty much stays the same. But these other isotopes, all the ones that have the large delta, the capital delta, 99, 01, and 04, they experience what's called mass-independent fractionation. And there's processes such as photochemistry in the atmosphere, like the oxidation reduction that happens from ultraviolet interactions. Those affect these other ratios. And so it's actually very useful to look at all the ratios and try to tease out what processes might have affected the mercury in its transport and its bioaccumulation. So our group in Wisconsin, headed by Sarah Jansen, has done a nice job of putting these slides together here about the mercury isotope results. This graph shows previous work before we started our study, but there actually were some samples from Sulfur Bank Mine published by Smith and others, 2005 and 2008. And our data from Sulfur Bank, their data, plots mostly in that red circle, which is pretty much right in the middle of all the samples from the coast ranges of California. There have been some studies of some of the other mercury mines in the area, and they have a bit of a range, and the horizontal axis is one of the small deltas, or that helps you understand sources, and the vertical axis is the big delta that helps you understand atmospheric processes and photochemical reactions. So there's pretty much a cluster or central tendency of that data in the red circle. So keep an eye on the middle of the graph. And this is, again, before we started our study, that was known. So then, with the help of technology, we will get to the next graph. Maybe. Okay. So groundwater, Carter mentioned there's been a lot of groundwater study at the mine, especially in that waste rock dam area. These are just samples from the very first year of that study. We've probably got another 2 or 3 times this much data coming soon. But it's pretty much in that same area. It makes it look like, the groundwaters get the same source of mercury as the mine waste and the ore. Maybe not surprising, okay? Then if we look at mine waste itself, most of the data points are in that cluster, but there's a few out there to the left and up in the top, which is kind of interesting. So which samples are those? It turns out if we plot those against mercury concentration and we use the same symbols, those are mostly samples of the calcine tailings, the ones where they've removed most of the mercury, so they have relatively low concentrations of mercury, and the south waste rock pile, which is next to the calcines, which actually has some calcines in it, we think. So they're probably not that important a source of mercury. The concentrations are lower. They have this somewhat different isotopic signal, which could be interesting for tracing mercury at the site. But when it comes to what ends up in Clear Lake, the action is really in these purple and green and blue symbols, which are pretty much clustered in mercury isotopes and also have higher concentrations. And so on the next slide, you'll see something about, should I wrap it up? Okay, Janine's saying wrap it up. So I'll go right to my conclusions here. Okay, well, biota are complicated, so I won't say much about it because there are other reactions, the photochemistry, the bioaccumulation, there's some fractionations that take place, and so sediment is also very similar to the other stuff. The biology is kind of complicated. We did see some differences in the lower arm that we don't quite understand yet in prey fish and zooplankton, but most of the biota, if you correct for all those reactions and what we call isotopic fractionations that take place when there's bioaccumulation and interaction with ultraviolet rays, it looks like they're all pretty consistent. But we've got a couple of anomalies to follow up on. We're going to get more data to see how real that effect is in the lower arm. But there could be some differences in the food web. We don't think there's a different mercury source, but it's something we're going to look into. And so my conclusions are coming, I promise. And if I don't get the slide, I'll just kind of wrap up by saying mercury isotopes are still preliminary in terms of our results. We have a lot more data coming. But it's pretty much confirming that the main source of mercury to the Clear Lake food web is Sulfur Bank Mercury Mine. There's a cool slide that you didn't get to see that shows that the air samples we took here in Nice and in Ukiah actually have different mercury isotopes. Oh, there it is. Perfect. Those background air samples have a pretty distinct signature from all these ones associated with mercury mining, both at Sulfur Bank and elsewhere. And so that helps us determine that there really is probably very little mercury in the lake that came from, coal burning in Asia, for example, or long-range mercury transport that you'd see in those background air samples. We think that this is consistent with the mine being the main source. And so I'll just wrap up by saying, USGS has been doing a wide range of studies on mercury in Clear Lake and at the mine site to support EPA's efforts, and a lot of it's a work in progress, but I'm happy to share the data we have with you today. Thank you.
So, well, thank you so much, Charlie. That is possibly the most technical, substantive science meet that you're going to get here at the conference. And for all of the scientists, we got to scratch our brains. Everyone else is probably scratching your brains in a different way. While Sergio's coming up and we're lining up the slides, I want to ask everyone, do you know what the most important environmental science text historically is? What's the name of the most important environmental science text? Silent Spring. Who wrote it? Rachel Carson. Where was her research based? Here in Clear Lake, on the Grebes. I'm seeing some eyebrows raised. Yeah, it was here, the Western Grebes. And so everything that Charlie and other folks are talking about, bioaccumulation, biomagnification, that started here. Western Grebes. We had a massive decline. Those beautiful birds, the Western Grebes that do the dances, the paradances along the surface of the lake, and they also do dances with pulling up water vegetation and showing it off. We had a massive die-off. Scientists looked into it, and it was Rachel Carson's seminal work that started it, and it was here at Clear Lake. So I'm going to ask everyone to be really patient and stay another 15, 20 minutes because we are feeding you. Okay, you don't have to go run away for food. Food is going to come to you. So if we can stay here at least until 12:15, 12:20, if you need to stand up at least to alleviate the rumpus, as long as you can do so quietly. And Sergio, muchísimas gracias por estar aquí. And please take it away.
Well, thank you, Jenny. Sounds good. Yeah, I think so. So hello, my name is Sergio, and I will be just talking about with you about the development of in-lake predictive tools for suspended sediment and mercury dynamics. Flower. Hello, hello, hello. That was not me. Is that better? Oh, okay. So again, thank you, Jenny. Thank you, Angela, for all the organization and everyone that has been involved. For everyone that didn't hear me, my name is Sergio Valbuena, and here I'm just presenting the rest of the co-authors of this work, and I will be talking about the development of predictive tools in the lake for sediment, suspended sediments and mercury dynamics. Right. I think this will be better. So a little bit of what we're going to go through. I'm going to be talking about what is entails about the numerical, like developing a numerical model, and what is the process that entails, and then I'm just going to go through different steps that we go through. I will be talking about the conceptualization of these numerical models. Then I'm going to give some examples, first for the suspended sediment model that we developed. And I will split that into the implementation, test, and validation. And then I'll do the same with the mercury transport and transportation, which is a continuous work that we're doing right now. What is a numerical model? A numerical model is basically a mathematical representation of natural systems in which we try to represent mechanisms to predict or understand different physical and biogeochemical processes in natural ecosystems. So we try to understand complex systems, and developing a numerical model goes through different steps, all of them being represented in circles in here because they are really cyclic. We might need to go back and forth to understand and to be able to capture properly and edit the different steps that we go through. So the conceptualization per se is where we define the physical and biochemical processes, the different mechanisms, how they interact between each other, and that basically the output from this is a conceptual map where we describe this connectivity. we move into the implementation where we basically use the conceptual model that we develop, and we start transforming this into mathematical expressions. We start analyzing the assumptions that we need to make in order to be able to implement this into a computer and through be able to use numerical methods to solve these equations and obtain a model that is capable of receiving some type of inputs and just give us results and in the form of outputs that predict the behavior of our natural systems. Then once that's done, which actually no one sees, we start showing the test where we analyze single processes and we start analyzing the inner process interactions of single mechanisms. This is just simplifying different systems in a better way. You will see examples in the next slides. But just to make sure that our model is working properly and that we'll be able to predict the results or like to predict the conditions in the real systems, in particularly in this case, Clear Lake. Finally, once we're satisfied with the test of these simple systems, we move into the validation step where we can be analytical from laboratory experiments, but also field observations or in situ observations. And we basically use what Charlie has been collecting and many other researchers have been collecting data in the lake and compare the numerical results with this. And the error between those, between that data and the numerical results is a statistical metric that we use to calibrate and make better our numerical model. So I'll start with the suspended sediment conceptual model that was developed at Turk. I did this as a previous step for the mercury model, and we needed to implement this to be able to characterize the mercury better in Clear Lake. As Carl mentioned and Charlie mentioned, the mercury can attach to the sediments. Therefore, we need to represent sediments correctly in the lake. Here what we're looking at is just the conceptual model itself, where we have the water in blue and the sediment in yellow, brownish. And the errors are just describing the different forces that are irrelevant to this model. So we have diffusion advections. That just simply means how we interpret the water motions vertically and horizontally in our domain. Then we have the gravity, because it's sediment, the sediment is heavier than water, and it will tend to fall to the lake bed. And then we have erosion and deposition. That basically tell us the mechanisms that are capable of getting sediment from the lake bed into the water column. And then the deposition is the mechanisms that brings the sediment from the water column and just settles down to the sediment to the sediment layer. Once again, this is just simple. We don't need to understand the complicated equations there, but it's just to give you an example of what actually goes into computer to be able to represent the mechanisms properly. Here I'm just showing again the arrows for the erosion and deposition. What needs to be known, sorry, what needs to be known in here is that the balance between the resuspension and deposition will definitely give us a knowledge if sediment is being lifted up from the bed into the water column. And both deposition and erosion, same as the setting velocity of the particles, are dependent on the sediment size and sediment density. And then if we have any sort of erosion, it will be because the water currents at the bottom of the lake are sufficient to pick up that sediment into the water column. So now we move, so this is just the mathematical representations, part of the implementation. And if we move into the test, like I say, there are simple cases, simple scenarios, and in this case we have a hypothetical case where we are looking here. In the vertical is just the depth, and in the horizontal axis is just distance. So if we're looking in this way, we're looking at a pool from the side, and where we're looking at the light blue with high concentration of sediment. So we have a pool that has a significant amount of sediment, and then the right panel is just showing in a line what we'll see the average concentration of this whole section. So a way to see it is just we have a pool with sediment. So what happens if there is no any water movement? What happens with the sediment? And this is just the result of it. So as we can see, because the sediment has a gravity, it will start falling down to the bottom all the way to the layer, sediment layer. It's worth noting that In here, I'm just representing the water column. Let me see if I can read. I'm just representing the water column. I'm not considering the accumulation that we'll see in a real system in the sediment layer. So we can see, because there is no water moving, the sediment just settles down until it gets out of our system. So that is just a simple test, and once we're satisfied with this behavior, we move into more complex systems. In this case, is the evaluation of hypothetical scenarios at Clear Lake, because that's our preferred lake. So what we're looking at here is two hypothetical scenarios. And what I want you to understand first, or to focus on, is the difference between the two of them. The one on the left is a clear lake in which there is no wind, meaning the water currents, the water is stagnant, is not moving. And the other side, we have clear lake with wind, with real meteorological conditions from the summer of 2020. What we see in gray line is just this cross section representing the Oaks Arm. And again, the colors in here is the light blue for high sediment concentration, and the rest of this, the dark blue, is for zero sediment concentration. So this basically just tells us a scenario in which we release sediment at the surface. So what will happen? Basically, on the left, we can consider it as the pool example, and on the right side is just the actual system. So if we look at the results, we can start seeing the difference where we see on the left side, we see, because the water is not moving, the sediment is depositing right straight to the bottom. In this case, we actually see the accumulation of sediment at the bottom of the lake. But it will focus on the right side and the actual conditions in our lake. We can see that the water currents start moving the sediment around all the way that is actually depositing all that sediment all across the Oaks Arm. And this is really important, because if we consider any sort of remediation strategies that implies releasing a sediment at the surface, we have to take that into account for the amount of sediment that we want to put in there. And this is just because if we are targeting a specific location, we'll have to account that the sediment is probably not going to fall in there because of the currents. So now if we move into a little bit, so in the previous was just deposition, right? In this one, what I want to show, yes, you're reproducing, is a clear lake sediment resuspension event. Again, these are for the conditions in which Clear Lake experienced during the summer of 2020. This is just a little time window of about 160 hours, but in the assumptions in here is that the lake bed, so we're assuming that our lake initiates with a material that is covering the lake bed of the bottom of the lake. So what happens if we have the winds that we experienced in the summer of 2020? Well, we can see from the videos, and I hope it is, maybe I have to reproduce again. Yeah. What we can see is that during that wind event, we can see that a lot of sediment was picked up from the bottom and lifted up all the way to the surface. And actually, this sediment that was picked up could actually be transported because of the currents all the way to the upper arm or the lower arm. And this is just an example of how complicated or how complex dynamics the currents are in Clear Lake, and how we should take that into account again if we want to do or consider remediation strategies that involve these sort of solutions of putting sediment on the bottom. So I talk about the suspended sediment conceptual model. Now I'm talking about the mercury conceptual model, which you have seen less complicated graphs for the conceptual model. But I want us to focus in here is just to see the complexity of all the processes that involve developing a numerical model. And the more errors that we see in here between all these boxes, the boxes are just the mercury species, are just the different processes that we need to account in implementing. And the more errors that we see in a conceptual model like this, the more complex it is to implement, the more complex it is to get it to represent the actual conditions of our lakes. And it's more likely that we'll need more data also to make sure that the model is behaving correctly. Okay, so again, similar to the same conceptual model. Once we have the conceptual model and after we implement that into a computer, I'm not showing the part of the computer because it doesn't make any sense. is just a test. We start testing with simple cases. In this case, we're looking, again, the top panel is just for elemental mercury, Hg0. The middle panel is for Hg2, and the bottom panel is for methyl mercury. In this simple case, we're analyzing how the concentrations increase because we're putting an inflow on the left side that is going to carry mercury with a velocity, and is going to carry all the way to the right side. So if we play the video, we're going to start seeing how the mercury it gets accumulated in the whole water column, and the bottom is just blue because this is a simple test, and the test didn't include the concentration and the transfer of concentrations from the sediment layer to the water column itself. So we can see as the flow develops, because there is an inflow coming in, we start seeing how the concentrations increases to a point that reaches a constant value. This constant value gives us some validation that the numerical model is predicting correctly. So if we move into a second test, this is again the same. Blue means, sorry, I didn't explain this before, and I apologize. Blue is low concentrations, and red is high concentrations of mercury. But again, in here we have elemental mercury, Hg2, and methyl mercury at the bottom. But in this case, instead of analyzing an inflow of mercury, what we're going to analyze is the atmospheric deposition of mercury into our system. As we can see from the video, the atmospheric deposition starts being at the surface, which gives us confidence that the numerical model is behaving as we expect, as the atmospheric is just like the top layer, and then it starts accumulating again, remains at the top, but it starts accumulating in like deeper layers. And the difference that we can see from the left to the right is just because there is an inflow of clean water coming from the left, and that's why we create this difference between the two ends of our system. Twice. And just to finalize a little brief of the summary and next steps, I just wanted to recall that we're building on previous numerical models that have been developed at Turk. These are the three main components. It has been developed a hydrodynamic model which has been calibrated or validated. So that gives me a step up because I'm confident that we're reproducing the water movement and the temperature of the lake correctly. It's also including the river flow dynamics, and then there has also been the development of the water quality that includes the nutrients, dissolved oxygen, and phytoplankton, which are important for analyzing mercury in a system. We have developed, now part of this project, this is a project that is funded by CNRA. The contract was signed at the end of 2023, but the project dates goes from July 23 to June 26. And the status right now is that we have developed the suspended sediment model as, again, like I said, as a pre-step for the actual development of the mercury model. We implemented a sediment layer, which wasn't a part of SI3D at the beginning. And we also implemented just the different mechanisms for representing suspended sediment in the lake. We developed the equations, so we did the conceptual model, and we developed and implemented the mercury cycling equations into the model. And we are in the phase that we're initiating the tests for different single mechanisms, and I'm increasing those tests in complexity as we speak. And I have also developed pre and post processing for being able to understand data, to input it into the model, and also understand the results from the numerical model itself. We'll be using USGS data from 2020 to 2023 to calibrate the numerical model. And then for the next steps is to validate and calibrate the numerical model with actual clear lake conditions, because at the end, that's what we care for. We care for this system, and we want to make sure that we're representing it correctly. And then there will be a model that will provide inputs into or insights into the biooptic to the lower food web. This will be a separate model. It's not going to be a part of SI3D, but it will feed up from the results from the model that we are developing. We'll be evaluating mercury dynamics under hypothetical hypolytic oxygenation scenarios. And just because of the timing of the HOS system program that you will hear later on, there is a probability in which, because of the timing of the projects, and this project will need an extension to be able to analyze in conjunction what is happening with the oxygen and what that affects on how that affects the mercury concentrations in the lake. Thank you so much.
Sergio, thank you for those graphics, those interactive graphics. That really showed how your models work. I would like another round of applause for our first panel session. You may have noted at the bottom of each slide that this is preliminary information subject to revision, not for citation or distribution. So keep it on the down low. For now, USGS does have very strict rules for their scientists about when and how and where they can publish their data. So if, Sam, if we could go to the schedule that's on the website, because as I've been noting, our incredibly detailed schedule that Angela spent so many hours agonizing over, in order to fit it onto 2 pages, it meant that the font is, kind of like Lilliputian size. But the schedule that is on the website, which is an overview, it's a 1-page overview, it's yellow and it's blue, and as soon as we get it up, I will be able to show you how it delineates each day. And that's an easier place to go to. You can pull it up on your phone, and you can then see where different sessions are. So for example, after lunch here, what we have are the permitting sessions, and then the Terra sessions on Indigenous knowledge, good fire by the Tribal Eco Restoration Alliance. That's what's going on here. Over at Habematolel meeting room, there will be the sessions on Clear Lake water monitoring, modeling, and hazardous algal blooms. And in just a moment, they're going to click up. There it is. It's that schedule. Aha. All right. So you see how there's Thursday on the left and Friday on the right? Morning sessions on top. For Thursday, everything that is in very light yellow, that's here in the Robinson conference room. Dark yellow, Habematolel. Tomorrow, light blue, here. Dark blue, Habematolel. And again, all you need to do is go to the Lake County Water Resources Department page, scroll all the way down to the Clear Lake Integrated Science Symposium tab, go onto that, and then you can click onto the program schedule, and voila. You see everything that we're doing. And hopefully we will have copies of this program schedule available tomorrow, because it's just easier to see. And then, if you want to get out your magnifying glass, you can then see the detailed list of speakers. So, sandwiches, are they here, Angela?
Yes, there's only 6 veggie on the left. Yeah. Okay, thank you again to our sponsors for providing lunch. We knew the time was going to be tight today, so we want to make sure you had that. There are only 6 vegetarian on the left-hand side. So if you're not vegetarian, please don't grab a vegetarian. And if you want something else to eat that's not over there, there's a cafe here. And there's also some other eating options. I believe Torped Out Kitchen is also a food cart in the parking lot. And I've heard it's delicious. Yes, I'm getting lots of nods. So thank you so much. We will break for lunch if we have nothing else. Okay. And questions directly to the speakers. Also today at the reception this evening, tonight is a great place to talk about this stuff, and there'll be posters, demonstrations. So thank you everybody for the morning session. We'll see you in the afternoon. We are still starting at 1 p.m. Thank you so much. Oh yeah.